This is ATP synthase complex. Some authors refer to it as ATP synthetase complex. Let me first explain the orientation for you. This is the inner mitochondrial membrane. This is the matrix. And this will be the intermembrane space. When I kept on telling you 10 protons have been held in the intermembrane space against their will, or 6 protons have been held against their will in the intermembrane space, I told you they are desperately looking for a chance to come back into the matrix. If they come back through multiple holes, energy will be dissipated without having any kind of use at all. But if you channel that energy through a small pathway, for example called as proton pathway present in the ATP synthase complex, this energy will be helpful for trapping ADP and PI together and convert them into ATP. This is where phosphorylation happens. We saw oxidation previously. Now we are looking at phosphorylation. Okay, now imagine this. Every proton that is present here will be able to push the C ring once. What is a C ring? For that you have to understand the components of this electron transport chain's ATP synthetase complex. What you are seeing here is F0 or FO complex. What you are seeing here is the F1 complex. Why is it called as FO? Because this complex can be inhibited by a drug called as oligomycin. So calling it as F0 or F0 is not ideal. Calling it as FO is ideal. This is FO portion from here to here. From here to here, it is F1 portion. So remember, FO portion is buried within the membrane. So FO portion is the most hydrophobic component. FO portion is hydrophobic. What about the F1? F1 can have moderate kind of activity. It can have hydrophilic portions also. Now, look at the F1 portion. You have an alpha here, alpha here, alpha here. So F1 complex is made up of 3 alpha portions, plus I have a beta here, beta here, beta here. So we have 3 beta portions, but there is only 1 gamma portion. Don't call this and this as 2 gamma portions. Technically, both of them are the same. This gamma portion has extended from here to here, and it has been covered by the alpha and beta and all the sides. Look at this. If this is the gamma, if you're looking at the gamma from the top till the bottom here, if this is the gamma, you have covering like this. In this covering, you can keep it like this. This is an alpha, this is a beta, this is an alpha, this is a beta, this is an alpha, and you'll have one more beta. So if this is the stack called as gamma, you will be having other proteins like this, alpha, beta, alpha, beta, another alpha, another beta. So you have alpha, beta, alpha, beta, alpha, beta in alternative functions, while the gamma has extended from here till here. So you will be able to notice the rotating portion will be this fixed portion called as FO. Now this FO is a drum here, right? That drum, imagine to be a circle in 2 dimensions. Look at the drum as a 2-dimensional circle. I have divided this into 10 portions, and each of the portion will be having a ring that is referred to as C rings. So we have C1, C2, C3, C4, C5, C6, C7, C8, C9, C10. It has been divided. So it means each of the ring has been divided 10 times. I mean, the whole circle has been divided 10 times. A 360 degrees has been divided 10 times. It means each of them will have a 36-degree angle. Now remember, when a proton drops on this area, this portion moves from here to here by rotation. Imagine this is the drum. If this is the drum being held like this, when one proton hits this area, then 36-degree movement can happen. So it means in the position where initially C1 was present, C10 will come and occupy, C1 will go to the position of C2. It means when 10 protons have hit, That is, each proton will move the ring by 36 degree. If at all you have 10 protons hitting, 10 times 360 degree rotation happens. So C1 will come full distance and come back to the same site. That is actually one rotation of the FO portion. Now what does this tell you? Here, this drum is rotating. The drum is rotating and you will be able to have this particular drum rotation creating stress on the gamma. Gamma will right now act as an axle which links this drum to that of the complex made up of alpha and beta. Apart from alpha and beta, you also have something called as delta, and then you have epsilon. Here you have the proteins called as A protein, which is actually making this particular drum bind to the membrane, and there is a B protein which is binding to the delta, which will be in direct contact with that of alpha and beta. Now the whole complex has come full circle. So if this is a membrane, the drum is bound here. Now the drum will be releasing a stalk. That axle is the gamma, and near the gamma you have alpha beta, alpha beta, alpha beta surrounding it. Now this is a mass. That mass is bound to a delta protein and epsilon protein, where you'll be able to have a protein called as B. That is again coming back and binding with the membrane with the help of a protein called as A. So the circuit actually looks like complete. That is, you start with the ring here, and then gamma axis or axle, you have the proteins here, and then you have a delta chain from which the B protein comes in, and then it is bound to the A protein, and the A protein is bound to the drum. So it is actually coming full circle. So you remember the contact between FO and F1 has been established using this gamma stalk. Now the rotation can happen in such a way the gamma stalk will try to remain as stable as possible, but because there is excess amount of pressure here, the gamma will be squeezed and slightly turned. Now you will be able to have the alpha, beta, gamma in their position. Because of gamma's rotation, with the help of the rotation from the C ring, stress will happen on the alpha beta positions, while the whole FO and F1 has been held in position using A protein and B protein, and the B protein making contact with the delta protein. This is the setup. Now look at this concept. This is called as binding change mechanism. This is called as binding change mechanism. What is this? An explanation for beta synthase or beta subunit acting as ATP synthase complex, because of which ATP is synthesized through the gamma axle rotation.